Well, hey AP, hope you're doing well when you watch this video. Welcome to part 1 in chapter 5. This is going to be about cell membranes and signaling. It's a great chapter. We're going to have a lot of fun with it in class, discussing various things that are like hot topic issues and caffeine and how different drugs affect the body, and medications, and all about communication in the plasma membrane. It's really impressive. It's very impressive. So without further ado, let's jump into Chapter 5, again, you're completing these notes based upon how it all goes. And so we're going to drag this up here. And so this is caffeine on the left and adenine on the right, okay? And adenine, of course, is part of different molecules, some cofactors, adenosine triphosphate, adenine. And so we'll talk about various things, and I'll talk to you about this stuff when we have class. But chapter 5 opening question is kind of this: what role does a cell membrane play in cells? And so, like I was saying, the chapter 5 opening question: what role does a cell membrane play in cells, and what is the one property of molecules that directly impacts their position in the fluid mosaic model? Very important to understand that key phrase and what it means, a fluid mosaic model. You can read through these key concepts. And so concept 5.1, cell membranes are fluid mosaics, okay? And so brain cells, I mean, here's an interesting example. So this is called a synapse, where one neuron connects to another neuron. And you have all these neurotransmitters inside these vesicles. And so you can see here that there's different types of stuff happening here and different things embedded in a plasma membrane and right here. And so that is an example of how brains communicate. And so I'll dive into this with you in class. Before beginning this chapter, you should be able to do the following: explain hydrophilic and hydrophobic properties of molecules. Describe or even draw the structure of a phospholipid. Describe the structure of prokaryotic versus eukaryotic cells, especially in the presence of membranes inside as well, organelles. Think about that. Basic idea of potential energy, and explain how proteins bind together. So, a membrane structure and function are determined by its constituents: lipids, proteins, and carbohydrates. The general structure is known as a fluid mosaic model. Fluid meaning things like float in a lake, and mosaic means it's always constantly changing and made up of different molecules. Okay, so here's a great picture that I would almost star and highlight that you need to know about. So inside this, diagram here of the fluid mosaic model, you have proteins. And so some proteins go across the entire membrane. So those are called integral proteins. So right here, they span the entire membrane. Some proteins are on one side versus the other side. So they don't span the entire side. So those are called peripheral proteins. Cholesterol is a big constituent in plasma membranes. It helps influence the fluidity of fatty acids in the membrane. It helps determine what is fluid and what's not. You can see right here that also is important is carbohydrates. And carbohydrates is about signaling. And so different carbohydrates on the outside of cells is kind of like an identification marker. It signals different things. And so that is an example of just some of the things. Proteins, lipids, carbohydrates, cholesterol, all involved with the plasma membrane. And so the plasma membrane itself is common to all cells. That's something that we don't say very much. And so the internal membrane, it separates it. It separates internal from external. The phospholipid bilayer, meaning two, okay? External surface with hydrophilic polar heads. So those heads are pointing themselves towards water. Oh, I'm trying to write, and that's not really working out. Those are pointing themselves towards water. And cytoplasmic surface lined with hydrophilic polar heads, again, because water is on the inside and the outside. But the hydrophobic part is sandwiched in between. I would start that. You need to know that and what it looks like, okay? And so this is hydrophilic regions and hydrophobic regions inside plasma membrane. So this is what a phospholipid looks like. You can see the hydrophobic tails saturated with carbons and hydrogens. And then you have glycerol and a phosphate, which is charge, right? You see the charges. Charge here. And then these are, these do have partial charges here because the oxygen steals more of the electron. Okay? And so, like right here, those have partial charge. So you have a hydrophilic head, which means it's polar, and a hydrophobic tails, phospholipid bilayer. And so the tails associate with each other because the hydrophobic parts want to stick together. So there's your sandwich. Here's your sandwich in the middle with the hydrophilic heads facing the water inside the cell and outside the cell. So there's three components in a plasma membrane. First off, there's lipid components, which is the phospholipid bilayer. There's proteins. So they float around. Fluid, right? Fluid mosaic model. This is the part of the model that is fluid. So fluid mosaic model. Membranes could be peripheral, which means on the outside, or spanning the entire membrane. And then cholesterol affects the fluidity, like how fluid the membrane is. Is it rigid or is it very fluid? And that depends upon temperature. Okay, we'll talk about that in a second. So most of the lipids and some proteins can shift about. They can move around. It's rapid. Proteins move more slowly. Lipids move very, very fast. Some proteins drift. They did a study with this, a mouse cell versus a human cell, different membrane proteins labeled radioactively, and then they fused them together, and after an hour, they were all mixed around because they could see, due to the radioactivity, the radioactive dye, where they moved to. But here's an important question, and actually objective kind of thing about. What happens with membranes when it gets really hot and really cold? So as temperature is cool, membranes switch from a fluid state to a solid state. Okay? So a membrane remains fluid to a lower temperature if it is rich in phospholipids with unsaturated hydrocarbon tails. Okay, unsaturated means there's like a little bend in it. But cholesterol, so let me just draw that for you. Saturated, no bend. Unsaturated, there's a bend. There's like a little double bond that causes like a little bend in the in the tail. Why would this keep it to remain fluid? Because when you lower temperature, kinetic energy is going down. And so if kinetic energy is going down, these molecules are going to become more tightly spaced together and could possibly freeze together. But if they're spaced out, that space allows them to remain more fluid. Steroid cholesterol has different effects. So at warm temperature, cholesterol restrains the movements because if they move too fast, the membrane could dissolve. And at cool temperatures, it maintains fluidity by preventing tight packing. And so cholesterol prevents it from dissolving here, doesn't allow it to dissolve here, and it prevents it from tight packing here, which causes it maybe to lose fluidity or even freeze. And so membranes must be fluid in order to work properly. You have to know how membranes differ in temperature. That is a learning objective. So you have to know how membranes differ when it comes to temperature. Okay, so here's an example, unsaturated versus saturated. That'd be very viscous. Packed together, increasing membrane viscosity. Not much fluidity there. The bent tails are help. Cholesterol reduces fluidity at moderate temperatures. But at low temperatures, it hinders solidification by disrupting the regular packaging, which is very important to kind of think about. And there's cholesterol and what it kind of looks like. You don't have to know what it looks like, but there it is. Two important factors: lipid composition, unsaturated or saturated, and temperature. And of course, cholesterol. So proteins, peripheral means on the outside. So they kind of anchor themselves on the outside. They're not embedded in the bilayer. Integral are partly embedded in the phospholipid bilayer. And so you gotta be thinking, proteins have a shape, right? Primary, secondary, tertiary, quaternary. And so these shapes determine if it has hydrophobic regions. So we would expect in this picture the hydrophobic regions of this three-dimensional protein to be here because they want to be with the hydrophobic regions. They want to have anchored within themselves. Fats like to stick together. The hydrophilic regions would be here and here. That's where we would expect the hydrophilic regions of the protein. Okay, that's just an example of what proteins look like underneath the computer model. Proteins are asymmetrically distributed, and so a transmembrane protein extends throughout the bilayer or integral, and so you can see right there the transmembrane protein. Some membrane proteins can move within, while others are restricted. How are they restricted? Because they can attach to cytoskeleton, and that would restrict their movement. Okay, so the next big learning objective for this section is functions of proteins. So lots of different functions of proteins. Channel proteins, carriers, cell recognition proteins, and then receptors, enzymatic, and junction proteins. Very important. You can see all the slides about this. You have to know these functions. Okay, these are easy note cards to make, not the easiest to learn, but easy to make. So number one, channel. We're letting things pass through a membrane. This channel is always open, so it's always letting passage through. Carrier proteins combine with it and assist passage. So these are not always open. They carry stuff across. Cell recognition includes glycoproteins. The term glyco means sugar. I'm trying to get a different color here. So this means sugar. Wow, that's not very good. And so it's all about recognition. We're trying to recognize maybe foreign cells, maybe our own cells. Your body knows you from not you, which is pretty impressive to think about. Receptor proteins bind. So these are like receiving. So we're responding to signals. Cells respond to stimulus. Receptor proteins allow them to do that. Enzymatic, metabolic, catalyzing reactions. And then junction proteins is all about attachment. Okay, so you definitely need to know all these 6 things. Transport, enzymatic, cell signal transduction, which is allowing the cell to respond, recognition, joining, and attachment. Lots of different things here. And here's the 6 kind of in picture form and a little bit more explanations. Cells recognize each other by binding to surface molecules. So membrane carbohydrates may be bound to a lipid, therefore forming a glycolipid, or a glycoprotein. Carbohydrates can vary between species and also even individuals in some cases, even within an individual's own cells. But here's the thing, carbohydrates are used for, well, that's weird. Carbohydrates are used for cell recognition. Carbohydrates are, okay? Cells have to know what they're dealing with. Foreign invader, friend, foe, they have to be able to respond to things, to other cells. Membranes have distinct inside and outside faces. So this allows arrangement, and what is this arrangement determined by? It's determined by the endomembrane system, the ER and the Golgi, and tagging these proteins for where they need to go. Oh, sorry. And so here's the endomembrane system. You can see all the lipids and all that sort of stuff. And so, for example, this just kind of walks you through how proteins are on various surfaces, okay? So it starts with the ER. Oh, I probably don't need to do circles anymore. And so you can see the ER. In the ER, carbohydrates are green. You can see the green right here. The purple dumbbells are transmembrane proteins. Secretory, proteins that are going to be secreted are those like purple circles. And you see right here, there's the transmembrane protein because it's embedded in the vesicle. You kind of just visually follow this. And so via vesicles, right? ER, vesicle, Golgi, tagged with different things, tagged where it's supposed to go. And then these are released. The glycolipids, are formed. And then they stay, and you can see right here where they stay. Everything right here. Oh, my bad. They stay where they should be. Okay, and that's just a great picture to kind of walk yourself through that. Okay, so we'll go through these answers to these questions in class, but definitely work on them at home. Carbohydrates are attached to the plasma membrane proteins in the ER. On which side of the vesicle membrane are the carbohydrates during transport to the cell surface? Propose a hypothesis for how the membrane lipid composition of a native grass found in very warm soil, hot springs even, would be different than that from a native grass found in cooler soil. We talked about that. Think about it, maybe rewind this video. And then finally, why are transmembrane domains hydrophobic? A transmembrane protein, why does it contain hydrophobic regions? Okay, this is section 1. I hope this was helpful. So proud of y'all. God bless. May the Lord bless you and keep you.